Oh, it was not in focus. So maybe
nice.
now it will work.
Yeah, that works. That's working? No. All right, so I'm Jonathan. I'm a software engineer at CDD. We are mostly based in California, but I moved back to France. So now we have a development team in Europe. I'm going to talk about hybrid molecules. So everybody is talking about this antibody drug conjugates these days. And we have a lot of clients asking us that, and we have a lot of issues on how to manage these. What kind of format are we going to use? How are we going to describe them? And that's the major issue. That has always been the issue with cheminformatics and bioinformatics, right? And if you have one thing to remember from this talk, it's this: is do not, when you import your client data, do not ever delete the data they imported. And that's a thing we have always done with mol files or whatever format they import in. We always keep it because we have to come back, either fix bugs or have new features, and they tell us, oh, we annotated that. We want to be able to use that. So we are always able to go back. The story of CDD, I will talk a bit more about it, but the story of CDD with RDKit is relatively recent. We were using a really large company I'm going to name before, and switch everything to RDKit for fingerprinting, searching, and all of that. And switching, we kept all the original files from our clients, so we were able to switch to the RDKit without any issues. So talking about bioinformatics, and especially peptides, proteins, DNA, RNA, you want to keep all the information, and you have biologists in your group. If you're a drug company, you have biologists, you have chemists, you have the informaticians, and everybody wants something different. Not being too far. Everybody wants something different there. Some of them want a sequence, some of them want a nice bead-like representation with colors. Some of them want to have the chemical structure and be able to search in the amino acid structure. So that's why we want a system that can keep all the information together. That may be a choice that not everyone will have made. Currently, we see a lot of companies are using Helm for storing their macromolecules. We decided to go with SCSR. One of the major reasons is you have everything in one file. That means that you have your structure, you have your templates, the structure with the different elements in them, and you have all your template elements, all of that in one file. You have the sequence that you can extract from the file. So that's one of the major reasons. A big problem with SCSR, documentation. It's mostly only Axelris and BioVia that made that, so no documentation. No software support when we started all of that. There was virtually no library, nothing out there to work with SCSR. But we did a survey of the different formats, and that was the one that would allow us to work the best. And the other reason is we are mostly mol file and more V3000 based, so that also made sense in that. What does it look like? If you are familiar with mol file, that's pretty much beginning of a classical mol file, but you have more information. You have class information, you have sequence ID and attachment. And the attachment part is the important thing. That's what's going to say that part is attached to that part. I'm not going to give too many details here. I know not everyone cares about technical details. And that's where things get different. Is you have new sections in your mol file where you're going to describe templates. The other big thing we were interested in, we have a lot of clients, they are going to import thousands of molecules, and they want to say, oh, I made that really special amino acid. We are the only one in the world making that one. And when you do the reaction, that's a living group. And that's the other main reason we are using SCSR, is that you can have the living groups, you say where things are attached, you can have many kinds of attachments, you can have non-standard attachments as well, because we have seen that too. So that's one of the main reasons we have switched that. What we have done at CDD is add the support for reading SCSR in RDKit. The reason why we want it in RDKit and not an external tool is that we want to compute properties, we want to be able to index them, we want to be able to search them. Another thing some of our clients are really attached to are the hydrogen bonds. Same thing, that's a confrontational issue for a lot of people. One of the things some are interested in are the wiggly bonds. So you have incomplete attachment between some partners when you have biopolymers. So a standard attachment will have 3 hydrogen bonds, but you can have it with 2. So same thing, adding methods to compute these and predict those. The way it's integrated in the application, they are going to insert the sequence. They can have a library of monomers. We display them. We try to do a nice view of them. One of the main features was that they wanted to be able to bulk import their sequences. So that's something we have worked on as well. So when they import things, they look at them like that. If they put their mouse, big advantage of not working with sequences or just with diagrams is that they can put their mouse on things and know that's the amino acid or that's the, we can do hybrids too. So you can have the details about every component of your biopolymer. For the chemist, they can always go back and look at the chemical structure. Of course, for cyclic peptide, it makes sense. When you have a giant protein, it absolutely does not make sense. You can work with RNA, you can work with double-strand DNA. And another thing we have worked on is antibody drug conjugates and antibody in general. I'm not going to be too close to that thing. Same thing, it's a giant mess. Nobody has a way to really describe them properly. Everybody is interested in different parts of the antibody. Some care only about the sequences, some care about what's attached to them, some care where it's attached, some don't. So we have heard a lot of different stories from our clients, and we wanted to have something that would cover most of the cases. One of the main issues is how do you display them? So when they are small, it's easy. When they start to get big, that's almost impossible to display them. Another issue is how you import them. That's something we are still figuring out and working on. Our probable goal here is to move them to SCSR, which is the other thing we want to add to the RD kit is the write support for SCSR. So you will be able to move things back and forth from the SCSR files. So for now, the clients are importing it with columns. They are going to describe the heavy chain, low and low chains, where things are attached, which molecules, so they can refer to them by their IDs or their structure. They can describe, some of them were really interested in knowing where the cysteine bonds were, so we added these as well. For now, that's the internal format we use, but again, that's the kind of thing we would like to use with SCSR and be able to move back and forth between the different things. That's just a representation. That's getting worse and worse. That's a representation, different representations people have for ADCs and antibodies in general. Same thing, there was no open source tool to do that. So we decided to do a system where we can annotate progressively and add all the information on the ADCs. Nothing to do with RDKit, I just wanted to mention that. But that's something we are more than likely going to do open source as well. And to go back to RDKit and CDD, we have one dedicated developer. Some of you in the room probably know him. He has been around for quite a while. He has added a lot of features in RDKit that we needed, Yes, of course, MRV, because we are still using MRV for everything that's related to reactions. So he did a lot of fixes for that. And some work on stereochemistry. There is still a lot to do here. In particular, it's pretty slow for us for some of our uses. You were talking, Greg, about slowness, and what Ted made is even slower, but it's more accurate in some cases. But we can't really use it in every case because it's way too slow. Atropizomers, that's a big thing that our clients were asking. So being able to describe your molecules in how they are adjacent in space and not just a stereochemistry. We use the Postgres cartridge, so that's our main way to register compounds. I can get further away too, but so that's the main way we register our compounds. So we use the Schrödinger layered fingerprint. Well, no, I think it's fingerprint, layered fingerprints. So we have a front-end service in Python that's going to calculate the layer fingerprints, just mash that up. We use it for substructure searching, similarity searching. We have hundreds of millions of molecules in the Postgres cartridge, so things can tend to be a bit slow. So we worked a bit on the queries to split things around, but that's something we really need to work on now because we are reaching some limits of, especially when we have simultaneous use, when they are adding things at the same time that they are searching, all the caches get wiped out. So that's a bit of an issue for us. Lots of lambdas, AWS lambdas, where we are cleaning up the molecules, calculating properties. So that way we can scale that up and don't have issues here. And we use a WASM JavaScript, the RDKit as well. So now it's everywhere. Now that we remove this old, really slow Java-based thing that we had before from a company, that works much better for us now. And we can scale, and we have been able to do more deployments, and we don't have licenses to pay. So our way to give back to the community is to have a developer that we pay and contribute things. So if you have anything in this stream that you want to work on or you have issues with, that's perfectly fine to come talk to us and talk to Tad and say, hey, I found an issue with that, or I would like to be able to do that, because it's more than likely that we would like to do it as well. So just come to us. So conclusion, what we see more and more is companies working on the same entities, and they don't want to use a lot of solutions. They don't want a solution for the biologist, a solution for the chemist, and a solution for the cheminformatician. They want one place to do everything. And that's what we are trying to achieve with macromolecules with the SCSR. Standard formats, I will not call SCSR a standard format, but we are trying to document it. We are trying to make an open library that's going to be able to read and write it with the RDKit. We try to open source things whenever we can. And we want to, and the thing is, when you work with molecules like that, if you provide all the information, or your client provides all the information, you can get all the functionality. But we had some clients that were just expecting us to store sequences and be able to compute things directly from them, or just display them. And then after that, they come and say, oh yeah, but I don't know where it's attached. And I have two different ADCs where I have different attachment points. How do I distinguish them? So that's a big issue for us to distinguish what's the same, what is not the same. And that's why we worked with atropisomers. That's why we work with tautomers. Everyone hates tautomers, but the clients love them. They want to say, oh, I registered it this other way. I know it's the same molecule. Well, so we use tautomers. We use fingerprints where we take preferred tautomer, so we can indicate to the client, hey, this is likely that same molecule as that one. But as you know, probably most of you know, that's a big problem. And with that, I'd like to thank you, and I will take any questions.
Also up, Jonathan. So do we have any questions? We have plenty of time for questions. Yep. So maybe Thomas? Yeah. You can repeat the question.
Yeah. Okay. Interesting how you integrate data from different protein and binding assays and the cell-based assays in your example.
So the question is about how we integrate assay data in general, so protein binding and all of that. Yeah. Mm-hmm.
By one assay, and you have to associate your technical profile in order to always show like activity, select this reference data for competition assays, and then that's very complex. If you add into that equation the cell-based assays, so for us that we start to accumulate data from NMR or the MSD from cell-based assays, and we have distributed it across different Excel sheets. And we're looking for the chemical sequence where we're looking for a similar compound bioactivity, because by physical assays don't always tell the truth. So you have to think twice or three times before you proceed with things from. So I would like to have one system where we can store all my data. And I would like to have associations between the results of different biophysical experiments and biochemical experiments. So I'm curious to learn more about how to handle that.
Yeah, so the question is about how to integrate your data, biophysical data, associating it, I imagine, with the protein and the target and the ligand. And I mean, that's more of a commercial discussion, so we can talk after with that, but that's the kind of things we do in the product. You can do mixtures as well. So you can describe that you have a mixture of ligands or a mixture of a protein and your ligands, and we are associating the bioassay data or assay data, really any data, to the chemical structure, to the you don't need the chemical structure, so we associate it to the entity. So your entity can be whatever you want. It can be a mixture, it can be a single compound, it can be a protein, it can be, so it's going to be associated with that. In the case where you have, for example, binding questions, that's going to be a bit trickier because we are not going to visualize the binding for you. So if you say it's between that site of the protein and that site of the molecule, you will be able to store it in the assay results, but that's not something we can display on a graphical way.
I want to go binding first.
Sure, I'm sorry,
Yeah.
can you maybe follow up and?
Yeah, I think we should, yeah.
Yeah, of course.
Yeah.
Yeah, all right, so
Good.
thanks again.
Yeah.